Hello, botanists. We are talking about propagation and genetic engineering and basically just how to duplicate plants. So let's start at the beginning. Cultivated plants originated from wild plants. For example, this is what a corn plant looked like. This is what a corn plant looks like now. We didn't do this with genetic engineering, just over time we picked the best corn, the biggest corn, the sweetest corn, and we went from this to this. This was just selective breeding. We just bred the one that was, we saved the seeds of the best ones from year to year. The appearance of cultivated and wild plants can be very different. You would not, teoscinate plants you would not know were the relatives of corn plants. This is the result of manipulation, genetic recombination, and there's a little bit of genetics. Yay! I know you guys are excited. Chromosomes. You should know this, your biology majors. The genetic information is carried on chromosomes. They transmit genetic information from one generation to the other. Some chromosomes are diploid, which is basically the vegetative parts of the plant, which is everything except the flowers and the seeds. The sex cells in plants are the pollen and the ovule. So think of the pollen as the sperm and the ovule as your ovaries. These are usually haploid cells. Chromosomes, again, review. Everyone should know this. This should not be new information. Long chains of DNA. DNA goes to RNA. DNA can replicate itself. You get two strands of DNA linked together. It's the backbone of the sugars, the deoxyribose, making it DNA, and it's linked by phosphates to each other. Replication occurs semi-conservatively, meaning one new strand and one old strand. There's a leading strand and a lagging strand. There's Okazaki fragments. This should all sound very familiar to you. This center part is attached by hydrogen bonds, so it can open up. C goes with G, A goes with T until you get to RNA, and then you have a U. It always bonds in the same pattern, G with C, A with T. Replication, the strands break, each base reattaches to its complementary base, and new identical double strands are created semi-conservatively. Okay, here we have again, here's your hydrogen bonds between your A and your T. Here's your phosphate backbone. This should all be extremely familiar to you. Here we go. DNA is the template for RNA. Now we're going to transcribe things. RNA is a single strand. It is the same as DNA, except it is single-stranded, and it has a uracil instead of a thymine. RNA is the template for proteins. Proteins are made from amino acids, and that comes from your DNA order. Then you go to your RNA template. Your RNA template transcribes. This is transcription. Then your RNA template is going to translate into a chain of amino acids, which will make a protein. Okay, now we have, this comes in triplets. AUG is the start codon. This is the entirety of the hereditary element. There are many chromosomes. Genes on the same chromosomes are linked. I'm not going to make you do linkage maps. They're usually carried together from one cell to the other. Here's a picture of a chromosome. Homologous chromosomes. Yes, sister chromatids. It's mitosis and meiosis, people. I know you're excited. 2N or haploid chromosomes. Diploid chromosomes, sorry, are paired. They're homologous. Corresponding genes on a pair are alleles, so big A, little a. Yes, this should all sound familiar. Alleles can be the same or different. So let me move this over here again. So you can have heterozygous, big A, or little a, big A, or homozygous. This becomes a little bit more important in plants because we're going to breed them, so we need to know who's homozygous so we can back cross them to someone else. Chromosomes again, dominant is one that expresses the alleles, big A, homozygous, dominant, big A, little a. Heterozygous, it's going to express this big A, but in plant breeding we need to know if it's homozygous or heterozygous. Recessive is only expressed if there are two little a's. This is a lot easier to figure out. So we can usually figure out what the recessive is in plant breeding. It's just harder to figure out the homozygous. We have to back cross them a bunch of times. Mitosis. Chromosomes. It looks exactly like animal mostly, except there are cell walls, which create this last step. There actually have to be a cell wall that happens there. New cells are 2 in pollen grains. Okay, this is an important little picture here because it tells you when you have haploid and diploid, when you have meiosis and mitosis. So essentially you've got your pollen grains here. That's going to happen with meiosis. Then once you have this generative cell tube, that's going to be mitosis. This is essentially going to be a little sperm in your pollen grains. That's going to go down through the tube, the pollen tube, into your ovule. Okay, so again, starting with the female part, you have your ovary. This meiosis is going to happen here. Then you've got mitosis because you have these cells that are going to divide. You have the one egg cell here, the pollen tube meets the ovary, and you have a little baby zygote plant. The reproductive division, better known as meiosis. Again, these are your homologous chromosomes. A line, one is from the male parent, the other is from the female parent. Alignment is random. Not really female parent on one side. We don't know that. That's just how we line them up to help explain genetics, but that's not really how it works. Your male chromosome doesn't line up to the left and the female to the right. That's not a real thing. It's just how we do it in textbooks. Chromosomes do not replicate. One goes to one daughter cell, the other goes to the other cell. Daughter cells are 1 in, a copy of one chromosome per parent. Many have combination of chromosomes from your grandparents. This is called genetic segregation. So we don't really know when these things start dividing which one of the sperm cells is going to mate with the egg cell. That's why you don't look exactly like your brothers and sisters if you are a twin. That's why sometimes you do get siblings that look very much alike, and you get siblings that don't look as much alike, because this process is random. Okay, let's talk about fertilization. The beauty when the pollen tube actually grows. So this is a pretty intense process, and it makes me respect plants a lot. So basically you get a pollen grain. It makes it to this stigma, and it's like, yes. Then it starts to grow down, all the way down into one of these ovules. And then when it finally makes it, this is released until you get the sperm nucleus that actually makes it into this egg cell, and you get actual fertilization of the plant. Here is your basic Punnett square. We're not going to do that. I'm especially not going to make you do double crosses with Punnett squares. You did that in genetics, so I'm not going to make you do it again. You just need a basic understanding. Genotype, phenotype. Again, genotype is the genetic material. Phenotype is the plant's behavior, its appearance. Plants don't really behave in a certain way, but they are resistant to disease and things like that. So we call that a behavior. Mutations. So we actually love a good mutation in plants because it makes things a different color or gives them stripes or makes them a little more interesting, and people like interesting plants. So we're all about trying to cause mutation. A lot of them go unnoticed. Some are basically delete mutations and the organism just dies. The plant just doesn't grow. But here's the important one. Some provide a source of desired variability for breeders. So all of a sudden something becomes insect resistant, or something becomes resistant to a disease. We love those. Or something becomes an interesting color, or does something unique. We really like that. So we're going to backcross it. That's where propagation comes in. Then we can just cross that one over and over with itself. Sometimes they can be induced. Yes, we induce mutations in plants. They don't even mind. We eradicate them. We put them in weird conditions. And sometimes you can get polyploidy. So you can get an increase in chromosome number. And you definitely get some crossing over in plants. And we encourage this because we like genetic diversity in plants because it helps with disease resistance. There's also some cytoplasm inheritance in the mitochondria and in the chloroplast. We like that to be variable because, again, it helps with diseases. Plant selection and breeding. Plant selection is the first step in plant improvement. So you notice the plant is hardier. You notice the plant stays longer into the winter, develops earlier, has a cool color. Just pick your interesting plant characteristic. Follows the sun better, has bigger seeds, all those things. You choose this plant with a desired trait, and you increase this plant number either by propagation, by asexual propagation, or by sexual. You can breed it back into a plant that's already working. Okay, next we will talk about breeding.